To this complimentary webinar, which is a co-presentation between Piramal and Sartorius. My name is Lennart Eriksson. I am part of the marketing team of Sartorius Data Analytics, and I will be the organizer and moderator for this session. And today we have the pleasure of having a title of the webinar, which is Design of Experiments for Early Phase Process Development of Antibody Drug Conjugates. This is a co-presentation between Xavier Despinoy and Eric Johansson. And Xavier is currently Process Technology Lead for Bioconjugation Services at Piramal in Grangemouth, UK. He holds a PhD in Organic Chemistry from the University of Edinburgh. After postdoc experiences, he joined Avecia, developing chemical processes for pharmaceuticals. There he started working on peptide conjugation processes with toxic warheads, which led into conjugation to antibodies, i.e. ADCs. Under CDMO Piramal, and since 2004, he has been involved in the development of many ADCs and bioconjugates. And over 40 of these ADC and bioconjugate processes have been scaled up to GMP and manufactured for therapeutic use. Xavier has extensive experience in technology transfer, process development, optimization, and robustness for scale-up and process validation. The co-presenter, Eric Johansson, is a principal data scientist at Sartorius Data Analytics. Eric has a PhD in organic chemistry from the Umeå University in Sweden. Eric has over 35 years of experience working with design of experiments, quality by design, multivariate data analysis, and multivariate calibration applications. In recent years, Eric has focused on modeling cell culture processes using batch evolution modeling and batch level modeling using SIMCA and SIMCA Online. And these applications have stretched all the way from PD all the way to the full scale manufacturing situation. And my personal note, knowing Eric for 3 decades, is that what Eric doesn't know about MVDA using SIMCA and SIMCA Online is probably not worth knowing. That was not part of Eric's bio. That was my little addition. So with these words, I now hand over the word to you, Xavier, to start this presentation. Please tell us your joint story.
Oh, thank you very much, Lennart, for this kind and complete introduction. And obviously the opportunity to speak today in a good company of Eric. So welcome everybody. So as Lennart said, we're going to discuss about a conjugation project using design of experiment, which has been carried out between Sartorius and Piramal, which started a few months ago. And I cannot move my slide. Oh yeah, here it is. No problem. So yeah, so on this project, Piramal have been working together, and Sartorius really bringing the antibody to the table, and most importantly, their expertise in design of experiment and the use of their own Umetrics MODDE software. And Piramal is bringing the drug linker to enable the conjugation and their analytical and process expertise in bioconjugation. So both Eric and I are presenting, but I'm the one to start. I will briefly introduce Piramal before discussing general aspect of ADC development. I will describe the ADC process we have been working on and talk about consideration for the application of DOE and selection of parameter and responses. Eric will then take over and talk in detail about the actual design of experiment, result, and the analysis. So let's get started. For those of you which are not familiar with Piramal Pharma solution and Grangemouth in particular, who are a world-leading conjugation contract development manufacturer organization with over 15 years experience in ADC conjugation and also in bioconjugation. We have capability on site to support services from the very early preclinical proof of concept project at low milligram scale up to GMP clinical and commercial manufacture. We have a very highly skilled workforce across a number of units, in particular the development, analytical and process group. I'm also pleased to say that Piramal has been recognized by the industry last year, the World ADC, as the best CMO provider for ADC. So Grangemouth is part of the wider Piramal organization, offering end-to-end services for a wide variety of pharmaceutical. But this slide is more specifically talking about the ADC integrated offering. We have capability in the US and Canada to manufacture the small solid payloads, which are the components which are typically used in conjugation. High potency API used in ADC can be made in Riverview. And we can also do a non-cytotoxic payload, which are made in Aurora. The center of the conjugation piece happen here in Grangemouth. And after almost 20 years experience, we have gained really a lot of knowledge and in both the development and manufacturing team. And we have scaled up so far, as Lennart mentioned already, 40 bioconjugation processes to GMP, including 2 now commercial products. The BDS produced in the UK can then be forwarded to Lexington facility in the US to become the DP presented as a liquid fill or lyophilized formulation. So let's talk now about sciences. So what are the goals of when you want to develop an ADC for the first time for human use? First of all, you want to have some very strong analytical method, which you need from the very beginning to start developing your process, ultimately to enable you to release your toxicology batch, GMP batches for release, and also stability testing, which are all required. Obviously, you need to develop a process which allow you to manufacture a product meeting specification. You want also to have some understanding of process robustness around the set point you're specifying in your batch record to ensure if there is any small deviation from the target set point, you will still meet the appropriate quality. And you also need to establish the control strategy. So before talking about the process itself, just to make sure everybody is on board, I just want to remind you of what we mean by antibody drug conjugate. Basically, an ADC, or antibody drug conjugate or ADC, is an antibody which has been chemically modified by a small organic molecule, a cytotoxic payload, or sometimes a drug linker combination, via formation of a covalent bond. The antibody is actually the carrier delivering to the antigen within the body the payload, which is the active substance. So in principle, the concept is very good. It's highly specific delivery of highly active substance, which should result in high therapeutic index. So it took the last few decades of development, but product has started to be approved in larger number nowadays, and to date we have 10 ADC approved on the market, with many, many more in all different phases of clinical phase. So in terms of analytics, ADC are based, essentially it's an antibody, so based on the characteristic of the antibody itself, biologic, but to which we add the additional complexity due to the payload. So some method will be very similar to those of the antibody. The one on the right-hand side column, for example, size exclusion chromatography, charge profile, ICIF, or SDS. Other methods are such as EEC or PLRP on the left-hand side, and free drug as well by HPLC, relate more to the characterization of the payload and its distribution on the antibody. All these methods are needed to develop appropriately your process. In terms of analytics, you also have other additional very key release assay, which are used to test the functional attribute of the conjugate. For example, binding assay, the cell killing assay, and the MAB specific effector function, such as ADCC or CDC. These methods are much complex, are involved in biology, and take much longer to develop. So are often developed in parallel to the process and not always available under the final format until quite late in the development phase. With respect to development of ADC processes, the effort can be summarized by the diagram here into 5 main stages with specific activity for each of the stages. That's the approach we follow at Piramal, and the approach has been designed to speed up preclinical program. In addition to those 5 activities, you got a number of other activities running in parallel. For example, analytical development, start even before process development start and continue throughout the development phase and even later for qualification activity. The formulation study as well can start before the active stage, but it takes a number of weeks, months, sometimes even possibly longer to get the appropriate final formulation. Okay, and the final stage before GMP is really you got a proving run at a relatively large scale or larger than what you have done so far to show your process you have developed, the formulation purification step are good prior to go to GMP. Okay, so the rest of the talk now really focusing on the design of experiment. So why do we use DOE? Eric could probably tell you more about it in a few minutes, but basically for us, it's a tool which facilitates the development activity and the planning of the experiment in a very efficient way. It allows a systematic assessment of the effect of simultaneously of multiple factors on a response. With respect to process development, the factors are the process parameter, and the response is specification or critical quality attribute of your product, of the ADC. This allows definition of a design space, which is really a safe operating condition for manufacture, which allow the specification to be met. So when and where do you use ADC? For early phase, we can use it to support both process and analytical development. For later phase, leading to commercialization, DOE become even more essential. It allows you to define proper design space, having a comprehensive understanding of the effect of your parameter and interaction between them, and also obviously define and allow you to rank the order of your parameter. Is that a critical process parameter or not? So how does a typical conjugation process look like? So you start from your antibody at the top, and the antibody might not always be in the right metrics to enable conjugation to be performed. So the first step is typically a buffer exchange or a simple pH adjust if the chemistry is compatible. Following that, you got the reactive stage, which can be in one or several steps, during which ultimately you're introducing your cytotoxic or non-cytotoxic if it's not an ADC payload. Then this is followed by a purification step, which can be chromatography or TFF or a combination of both. And if you have chromatography, you will follow that by TFF, buffer exchange, which lead into formulation, which is a stage at which you add all the necessary excipient to ensure you get your product in a stable state. And then you also perform funnel filtration, sterilizing filtration. You end up with your conjugate in an appropriate container of choice. So for this particular example I've been working on, here is a chemistry which was actually used. So you got 4 steps for the reactive stage. The first one is, as I was saying, pH adjust step and also dilution in order to get to targeted concentration and appropriate pH for the chemical reaction to happen. This is followed by the reduction of the disulfide bond, intramolecular disulfide bond of the antibody to generate thiol. The next step will be conjugation of the payload onto the thiol generated at the previous step, and usually that payload is used in a solvent. And finally, you also got a quench step, which really quench the excess of payload you might have introduced to ensure complete conjugation. So in terms of factor and responses, now you need to think of for the design of experiment. There is a large number of potential factor or parameter which you can pick for the reactive stage. So I've listed a few here, protein concentration, pH, temperature, standard one, number of equivalent of your reagent, T-cell or payload, the reaction time, the percentage solvent. You could add some more. In terms of responses, again, you look at your ADC, what are the critical attributes of your ADC, what is important is really to select the CQA which are going to be affected by the step, by the actual reactive stage. So based on that, you can narrow both columns, number of parameters and number of tests you're going to perform. So for I would say for an early phase, in terms of responses, you probably will be looking at aggregation and the drug load as a minimum. So just to explain briefly in a bit more detail the concept of drug load, you need really to understand the chemical reaction. So you start from an antibody, you got 4 disulfide bond for an IgG1, you reduce it, which will activate the antibody by generating the free thiol. So you can get up to 8 thiol for the IgG1. And once the MAB is activated, then you can add the payload on each of the activated site. What you get is not a single molecule, and it's different from organic chemistry, small molecule, but you get a mixture of conjugate with different number of drug per antibody. You can have 0, 2, 4, 6, 8 drug per antibody. And also you also got positional isomer for an analytical number of drug. So how do you characterize the distribution? So hydrophobic interaction chromatography is the analytical method really, which can allow you to look at the distribution of the different conjugated species as a function of their number of drug. For this product, so we perform IC, analytical chromatography, and you can see the distribution, which is on the left here, with a separation achieved for the different conjugate bearing either 0, 2, 4, 6, or 8 drug. Based on a peak area, you're able to calculate an average drug load for your BDS, which is one of the critical quality attributes of the ADC, which is also relating to potency. The DAR is really the response we're going to be discussing in more detail in design of experiment. On the right is just a picture of a UV spectrum overlay of each of the conjugated species after normalization relatively to the antibody absorption, giving a quick confirmation of the peak assignment. When you start a project, it's quite handy. So how do you get started, and what do you need to do before performing a design of experiment? So again, technically sound analytics for the response selected are critical. Sound may be obvious, but if you have too much variability here, you are at the risk of not detecting potential real effect or losing some resolution and only being able to identify the very strong effect. You need to select the parameter and the range. So selection of parameter is based on a number of factors, but prior generic knowledge of similar type of processes is very important, which you can complement by specific product knowledge if available. If there is no specific product knowledge available with this particular antibody you're working with, you can quickly evaluate some of the parameters and their ranges by doing some scouting experiment. There is also a number of statistical design available to use for DOE. Selection is guided already by a number of parameters, the resource you have at your disposal, and information you want out of the design. For early phase, we are typically using factorial design, but I've used some other design as well. Once you have a selected process parameter and a design, there is still, in terms of lab work, a lot of preparative work before you can actually carry out your design. For example, if you look just at pH or concentration as variable factor, how can you generate the appropriate representative feedstock material? You have either pH adjust or buffer exchange. So again, you need to think about that. What scale are you going to perform a DOE as well? So you need a model here. And depending on the model you choose, the model itself, the scale might introduce variability. So you need to have an understanding of the system really you are using. So this slide here is showing you an example of a scouting experiment carried out very early on, and it's probably, it will be the first experiment carried out with this antibody here. What we do first, again from experience, is we look at different TCEP to antibody ratio and also as a function of time. Give us an idea of how efficient is a TCEP to generate a number of thiol and time for the reaction. You can then plot the DR versus the TCEP, which show the linear relationship on the right-hand side. So if you target a DAR of 4, for example, you know how much TCEP you need. With respect to time, so for this particular DOE, we decided to keep TCEP fixed. I think we used 2.2, something like that. With respect to time, we retain that as a process parameter for the DOE, but we focus on a shorter reaction time as the reaction appear to be complete pretty quickly from that screening. So here is the selected parameter for DOE. So in addition to the reaction time, we have selected process protein concentration, temperature, and pH. Again, based on the potential quality impact and supporting data that you need to cover potential variability when you go to manufacture. Temperature is as part of your control, pH, concentration as well, dilution. On the right-hand side, we put some typical control ranges which are used in our manufacture. The concentration, so the range as well are shown. So concentration range, it's a good idea to keep those range pretty wide when you start initially if you don't know anything about the process. as it's beneficial, for manufacturer to work at highest concentration, high concentration in terms of productivity. It might not be so key when you start a process for phase 1, but when you start scaling up your process, to several hundred grams or even kilogram, it will make a big difference to your action vessel. And it's also good to look at lower concentration range because they can have positive impact on your aggregation, for example. So unless you know a little bit about your process, keep it wide. The pH, again, as well, can be kept wide initially. So typical control on plant is standard, I would say, plus minus 0.2 pH unit. So you want to look at something wider than that. Okay, and the final thing as well, once you have selected ranges, particularly if you don't have many information before, it's really important or a good idea at least, to test the extreme of the range selected for an early phase, or if you had very little knowledge, you will expect to see some variation there. So when we did that work, we got for all the parameters at the low setting, we got a DAR of 2.9, and for all the parameter high setting, 4.1. So we had a sufficient range of variation. In terms of size exclusion, so we did perform size exclusion, but we're not presenting the work here. We didn't see much variation at all throughout the entire range, so we had no issue with respect to aggregation, for example. So that's why we focus on the size exclusion. And that's where I hand over to Eric. Eric?
Thank you, Xavier. We will now try to shift and make Eric the presenter.
So first about MODDE 13, and a lot of text here, but basically the important thing is that the MODDE 13, you get the new optimization wizard that complements the reworked design wizard and the updated analysis wizard. That is the strength of MODDE. It helps you to get through all of these things in these design of experiments. So factors, responses and factors, that has been discussed already by Xavier. And you can see that he wanted that the DAR should be between 3.4 and 4.4, and ideally it should be 3.9. We have the factors settings that was also shown earlier, and that then, so to say, gets you into the design wizard that you see up there. I should pointer option, laser pointer. So that is from the design wizard. Here we have a full factorial design with the worksheet, and this is 16 experiments with 3 center points, and that will give a certain model. This is also standard and comes from the design wizard. What is really, really important and was really good that we discussed is that here you see that we have the performed experiment, not the ideal experiment. Everybody that's done experiments, it was a long time since I were in the lab, but I know that it's very hard to get it perfect. So you have to put in the real values here. And there is a way to test this, and that is something called the condition number. And the condition number will increase a little bit when you put in the real values. But the problem is if you work with the theoretical values, then you don't get the correct result. So you have to put this, and this is the problem in most, so to say, published examples. They show the theoretical values, not the real values. Then we go to the analysis wizard, and that's a light bulb, and it shows us here that here we have a really good chance to get what we want. First of all, we see that only 2 experiments, these 2 that are outside the red lines, they came from the definition. We also see that the center point is the replicates that are the center points are very, very close to the target. That is really, really good. And that's shown that the scouting and the knowledge has been strong. We also see that the model is very good. So everything is going well for us. So with this kind of data coming in, it's very easy to be a do my job. You usually look at the histogram, and ideally it should look at the Gaussian curve. And this is not a Gaussian curve, it's kind of skewed. But it's slightly skewed, and the model is good, so we don't do anything about this. This is also the next step in the analysis wizard. Then we get into the coefficients, seeing what influenced the model. And here we have the main terms, and here we have the interaction terms. And this one is definitely not significant. So therefore we can take it away. And you see that maybe we should take this one away, and maybe this one also. We have a very nice, so to say, feature in the analysis wizard called auto-tune or one-click analysis that does this by automation. And the advantage with the automation that all of us will get the same result. So that is the strong part of the analysis wizard also, that we don't have to look into them one by one. We can also go do the auto-tune and it comes out the same for everybody. And we continue here with auto-tune only, and we don't take away anything more. So this is the model that we use. Auto-tune will look at this, so to say, parameter called predictive R-square. And as long as that is going up, then we have it in. If it goes down, we go back one step. And then we look at the model. Ideally here we should have 4 thumbs up. This is how well the model is. Fits the data. This is the predictability of the model. Should ideally be as high as this one. Reproducibility should be as high as possible. And this is really, really good. And model validity is kind of low, but that is because Xavier did 2 good experiments. So this is basically a statistical artifact, because when this one is brilliant, this one will go down. So nothing to worry about in this case. And here we have, so to say, the model here. And apparently I have got help, then I should have fixed that with points instead of point. But you see that this is the really, really good model. Everything is very close to the line. If the R-square here is 1, everything is perfect on the line. And let me say then, when you do design or experiment and you get the model this good, you will be very happy. This is another very, very nice tool in MODDE, and that is the 4D contour plot. This overlap plot has been added to the analysis wizard. And we can see that the target is here. We have the factors here. And we have the targets here. And we can see that this corner is not good because here we are below the minimum. And also this little corner is not good. Then we go into the design space that is part of the optimization wizard. And now you can see that now it's not everything is green. And the reason is that here we see that, okay, what is the risk of failure here? This is something that you set yourself. Here we said we accept a risk of failure of 5%. and everything is green, then the risk is less than 5%. We see that we have more red when we have higher risk of failure. So that is, so to say, how we do this. But this so to say, design space is a really, really good thing. And when it's plenty of green, then you should be plenty happy. Then we calculate the robust set point. And the robust set point is the point that is furthest away from everything that is green. Now you have to realize that we have, so to say, a 4-dimensional space. So it is a bit of difficult to really visualize things in 4 dimensions. But this is a very, very good way to do it. So here we have the robust set points. And here we have a favorite set point. The favorite set point is the set point that you choose yourself. And it can be done because it is, so to say, more economically better. It can be easy to have the more even numbers or other things. And of course, it is very good if your favorite set points and your robust set points are very close to each other. That makes life much easier. So then we have summary in numbers, and that is also very good. Because here we have the robust set points. And this is the practical set point that we arrived with together with Xavier. He wanted to have the protein concentration a bit higher, and that was related to this NOR that he wanted to have a bit higher protein concentration. This is just easy to 20 centigrade, easy to understand. Here we have another pH, why we changed from 32 to 33, I don't remember. And the reduction time was slightly shorter here. But basically, this practical set point, you can choose yourself. That is just up to you. Down here, we have something interesting, and that is called the hypercube. And I will try to explain what I mean with this in the next slide here. We stop there because the hypercube becomes a bit too many slides, I realized. This was something that we did together, Xavier and me, but of course, that we should say that in summary here, that it's not common to get a large design space from a screening design. Usually you have to do an optimization design first. Why we got able to do that, we had excellent chemical knowledge that supported the selection of appropriate factor ranges. And also just this bookkeeping, realizing that we should enter the correct values in the performed experiments worksheet. And I would claim that 90% of you see in the literature, they have not done this. And I also have worked, so to say, on the dark side, working with authorities, and I've got these perfect designs. And I then asked the authorities, send back a question, did they actually do this experiment, or did they do other experiments? And then comes the answer with the correct data. And then the authorities seem to be a little bit upset that they got the incorrect information to start with. And of course, you have to have excellent execution of the design with very small replication and no noise and no outliers. That makes it much, much easier to do the MODDE part of this. Acknowledgement, of course, it was very much Xavier and his people that did this. And on the left side, we have seems that people that should be acknowledged. And also, of course, from the Sartorius side, we have a number of people that also should be acknowledged that helped out in different ways. So thank you all for that help, because that made it possible for Xavier and myself to do this presentation. And here we have the contact details for both of us. And then I have ended my presentation, so we can look into the questions.
Thank you, Eric. We have several questions. I think the first one is for Xavier. It relates to the fact that you showed this HIC diagram. Is that the best method to determine the DAR of conjugates?
It depends. You don't always get the nice profile I show you. For that, you need a good payload, sufficiently hydrophobic, and also the right chemistry to achieve that level of resolution. We found that the HIC is more applicable to the cysteine chemistry, we presented here, or site-specific Cyomab type technology. Lysine is chemistry is more heterogeneous, so probably too complex to get a DAR out of it, but you can still use it potentially HIC to get residual unconjugated antibody. Yes, there are a number of other methods, we can be used to determine payload to antibody ratio, DR, which our team regularly evaluate as orthogonal method when developing the actual DAR assay. It can be as simple as UV, but you also have HPLC. I think there was a trace, what we call PLRP, which is a reverse phase assay after reducing the MAB. And obviously you also got mass spectrometry as well.
Thank you. Then there are two follow-up question. What's the ideal DAR ratio? Can you comment on that? Do you have any target DAR that's always the ideal one?
No, they are not. You'll see classically, the cysteine chemistry, people were always starting with 3 to 5, but really it depends on what's happening to your conjugate in the body. Do you get into the right PK? Yeah, there's a lot of data to target. I can't really comment, but yeah, you see 2, you see 4, you see 8. There's a bit of everything out there. So know your target. It depends really on the payload as well, how much, if it's very hydrophobic, your molecule might not be able to tolerate as much payload.
Okay, then there are questions that I think, they touch upon each other. And one or two people are wondering about this scale down model, if you can comment a bit more on the impact of it, and then also what volumes are your scale down models working with? I'm combining a couple of questions here.
Yeah, so a scale down model can be anything really from working in a small Eppendorf, 1 ml to working in a small vessel of glass vessel, 50 to 100 ml. What is important is to understand what you're working with, potential pitfall of them, which could, if you introduce random variability outside the effect of the parameter you're looking at, it could ruin your DOE. So you don't need necessarily to have qualified model, but you need to think a bit of what you are using, what can be different at scale versus your scale-down model. So depending on the size of scale-down model you choose, you might want to perform some quick check, which are specific to the molecule you're working with. In this antibody we have used with Sartorius, so we were working at 1 ml scale. We found that when we were adding the payload, obviously mixing, if you are not mixing quickly enough, that could lead to aggregation. So there was quite a sensitivity here. So mixing is a typical parameter you probably want to investigate in again and 2 or 3 small-scale experiments to understand if it will affect some of the CQA you're looking at particularly during key addition, and how quickly do you mix, or do you mix during addition? Where do you add your reagent as well can impact, for example.
Thank you. Next question is for Eric, and I think you touched upon it. You here calculated the design, or the design space is usually calculated using an optimization design, but you are using a screening design here. Why? And can you give an explanation?
Well, I mean, the beauty with factorial design is with 4 factors. You can start with the screening design, and then you can run the full factorial. And if the full factorial gives a good result, there's no reason to go further, because resources are always scarce. So getting up to an optimization design, that would have needed 10 more experiments, and that would be hard to schedule.
Okay, thank you. Then there is a question on one thing in the analysis wizard. If the response, and I think it's then the histogram, if the response is not normally distributed, how can you deal with that?
Well, if it's, so to say, skewed, like Austria or Norwegian ski jump, you usually try to do a log transform. But when that's a positive skewness. When it's a negative skewness, it is not easy to get it in a good way. But a positive skewness, I usually try to see what happens if I use a log transform of the response.
Okay, thank you. Then one more in the analysis wizard. You commented on the yellow bar, the low model validity, and the other bars that were high. And you mentioned that a low yellow bar is sort of a more cosmetic issue than a real problem. But the question is, can it be the other way around, that you get a very high model validity but low reproducibility? And if you can have that, what would it mean?
Yes, that also happens, because when you have the way it works, when you have a low reproducibility, it's fairly easy to get the high, so to say, model validity. But when you have a low reproducibility, the R square and Q square will also be low. So if you only have the yellow one good, then you're in trouble.
So there are multiple options here revealing a weak model. Now it's over to you again, Xavier. A couple of questions that I will combine. One is how much ADC is required for a phase 1 trial? And then also, can you comment on how to initiate DOE for novel ADC linkers on an academic lab scale?
Sorry, can you repeat, Lennart? Sorry, the second question. Sorry, I didn't get that.
Yeah, I combined them, but maybe let's take one at a time. How much ADC is required for a phase 1 trial?
A phase 1 trial. If you're talking about the clinics, it will be very dependent on both the antibody and the payload. How potent is, how much does the antibody internalize. So we do see a wide range, but for ADC, what have we seen? We have seen as low as maybe 30 gram, maybe 30, 40 gram, and higher scale, a few hundred grams. So yeah, typical program, I guess, for phase 1 is, I would say, typically 1 to 300 seems to be the sweet spot currently for phase 1.
Mm-hmm. Thank you. And then the other question was, can you comment on how to initiate DOE for novel ADC linkers on an academic lab scale? Can you pass along some experiences or why some wisdom?
Yeah, so again, it depends on the chemistry. There is, I would say, yeah. What do you say? In terms of parameters, there's some parameters which are applicable to all type of chemistry. You have seen the pH, temperature, concentration are usually factor affecting affecting your chemistry, particularly pH and concentration. If you have side reaction, you need.
Okay, thank you.
So yeah, maybe we could follow that up. I think it's difficult without.
Yeah, we can definitely do. The question again for you, Xavier, if you do not have standards for DAR analysis in HIC, what would your approach be to identify specific peak for DAR?
Yeah, so what you can do, you can actually, if you got a separation like we did get, you can actually collect the peak you're getting from the analysis, and then those peak you can submit them to second analysis. You can either do straight mass spec, or you can also reduce the individual peak and analyze by PLRP, as you were saying, for example, and that will give you another measure of the actual peak. So you'll be able to say what is the DAR2 by mass spec and by another HPLC method as well. So you could, just the HIC itself, if you get a good separation, collect individual peak and then carry out multiple type of other analysis, PLRP and mass spec, for example, two independent one.
One more for you, Xavier, then I think it's for Eric, but this one is for you, Xavier. Apart from the conjugation process or conjugation step, for which process or process step can we apply DOE in ADC? Can you comment on that?
Aside from the reactive stage, you can use DOE for everything. I mean, TFF in particular, you get interaction between factors, so it's very good to do. Everywhere we need a bit of robustness, so typically we apply them to reactive stage, like if it was system chemistry, reduction, conjugation. If you want to do it for quench, you can do it as well for quench, for any chemical reaction, and purification, chromatography, TFF.
Thank you very much. There are coming in a few more questions. I think we have one more for you, Eric. Then we are approaching the hour here. It deals with this, again, going back to the model validity and lack of fit. How do you deal with outliers if lack of fit is significant?
Yeah, the outliers is always a problem. I mean, it's very easy in the model software to remove them. That is easy. Ideally, you should, if you have an outlier, you should redo the experiment. If you can do that, that's the best way to do it. Secondly, you can, so to say, remove it. If the outlier is by far the best experiment, then of course you should repeat it to make sure that it is the best experiment once again. There are no real rules. There are no statistical rules how to handle outliers. It's very much a discussion with the person that done the experiment to try to figure out what went wrong. Is it the experiment, or are we outside the limit where the experiment will work? So that is a difficult thing, and that is when I have to go back to the person that knows the chemistry or biology.
Okay, and then the final question here is for you also. If I want to have a demo of MODDE, who do I contact? Can we have a demo in MODDE?
Definitely, you have a lot of people that I've listed here. My name is also a person to contact. And also you can download the MODDE for a trial MODDE from the web if you want to do that. For 30 days, it's a full version of the MODDE software.
Yeah, I think also in the follow-up email coming after the webinar, there will be a link to a landing page for download. Okay, there is one or two more questions, but I think we have to, there are some things we'll need to counter questions to ask to fully understand the scope of questions. So we will leave those questions and follow up with them outside this session. So with that, I would like to thank both Xavier and Eric and all the watchers. But before we close, my usual thing is to ask you, Eric and Xavier, is there anything that you have come to think about that you should have mentioned? that you forgot to mention on a slide or something? Any final word?
I hope so, not.
Any one of you?
I hope not from my side.
No, I think for me it's useful to have a statistician to speak to if it's the first time you are using DOE. But so Eric input was very valuable.
Yeah, but I mean, I'm not a statistician. I'm a data scientist, but we can help you.
Sorry.
No problem, no problem. But basically, if you have the MODDE software and you are worried, just contact us and we'll help you. That's not, we can set up a short webinar. All of this has been done by short webinars.
Okay.
And we learn also quite a lot. So just contact us and we will help you. And this help is free of charge. After 10 questions, 10 times the webinar will start to charge, but not before that.
Thank you, Eric. We take that as the final words here. So from my horizon, thank you, Eric and Xavier, for the most inspiring and instructive webinar. To all the watchers, thank you for your time and attention, and I hope you will find this presentation useful once you get access to the recorded version. And with that, I think we close here. Thank you very much and have a continued nice day. Bye-bye.